Welcome to the Renal Research Institute's Frontiers in Kidney Medicine and Biology, where we share knowledge and advances in kidney research with the world. In this episode of Frontiers in Kidney Medicine and Biology, we talk with Dr. Alhaji Cherif, Principal Mathematician at the Renal Research Institute. We discuss COVID, chaos, and computation, what mathematics can tell us about pandemics. Dr. Cherif has trained with some of the world's leading mathematicians, and he is bringing his expertise to projects as diverse as COVID-19, bone metabolism, and anemia management. It's such a great pleasure to welcome today to Kidney Medicine and Biology Dr. Alhaji Cherif, who is an esteemed mathematician. He is specialized in applied mathematics. So welcome, Alhaji.
Thank you, Peter. Thank you for having me.
No, my pleasure. So, Alhaji, you are an applied mathematician. Just help me to understand this. I mean, we all have mathematics in school, but there are these applied mathematicians and the pure mathematicians, and it's another species of mathematicians. So what's actually the difference there?
So applied mathematicians usually tend to deal with real-world application. So we basically write equations to describe the real-world problems, phenomenon, and trying to model them by equation formulas to better understand them by managing them or predicting them more efficiently. And pure math, in a sense, it deals more with abstractions and deal with abstract entity and try to find relationship between these different abstract entity. In a sense, pure math try to understand deeper the mathematics itself, whereas the applied math trying to apply some of those tools in more real-world cases. And you might even be familiar with this. So whenever you go to your doctor and your doctor write a prescription for you, in a sense, they're using equations to give you the dosing that you need for your prescription. So in a sense, by not directly telling you, that's what they're doing. So that's what applied mathematics is.
Interesting. I try to recall when my doctor, I myself, would use a calculator the last time when prescribing something to a patient, but I guess you are referring in the background, say the pharmacokinetics, pharmacodynamics, exactly. They will all evaluate this. Which actually brings me to the next question, Alhaji. How can applied mathematics actually help patients? I mean, do you have a few examples where applied maths really made a difference?
Definitely. So there are a lot of areas that applied mathematics has been useful. So like in the medical world, like your CT scan, right? It's actually driven by a lot of mathematical models. But in recent years, mathematical models have been used where we use it to evaluate the effectiveness of new drugs or new procedure. We use it to, let's say in the cancer world, you look at it, they use it to estimate the survival rate of cancer patients undergoing a certain type of treatment, like glioblastoma example. Mathematics has always been used there in conjunction with computational tools to understand the growth of cancer and how to control them for more than 2 decades, right? So here is mathematics allow you to understand the challenges in cancer treatment, including drug resistance and relapses. And in my own work, we use mathematical model in renal anemia, where mathematics is used to provide administration dosing to patient. Or mathematics can also use to understand some of the pathophysiological processes that may not be fully understood, but allow us to understand some of the intricacies of these particular fundamental processes. So mathematics can actually apply mathematics in this context can be used a lot in helping patients improve their health outcome and also have a better treatment protocols.
Yeah, this is actually really interesting when you think about it. Say you go, you have lab tests done, you have an imaging done, you get drugs prescribed. That on so many levels, mathematics is really involved. It's like I don't know, using a smartphone and you don't realize how much math is actually embedded there, and nothing would work without mathematics. So
exactly. Yeah. Even if you do like a Google search, right? Google search have a page ranking, so that's a mathematical underlining. Or if you get recommendations on YouTube or other site, like when you're shopping, all of these have mathematical underpinning, and they might come with different names. Some people might call it machine learning, and sometimes it's just mathematical model based on data, analyzing the data that you're generating.
I mean, in these days, now in the midst of 2021, it's very difficult to talk about anything without talking about COVID-19. And I know that you are conducting active research into some aspects of COVID-19 and how to describe those aspects mathematically. Can you give us a few examples? I think you published about pool testing, you published about the reproduction number and other work. So can you just give us a few examples and tell us how you approach those?
I work on several mathematical models related to COVID. So one of the projects that I'm working on is analyzing actual COVID cases among our patients using within a host immunological model to understand how our patient responds to infection or even to vaccination. And this can be used to predict which patients are more likely to need additional vaccination or how soon they will need additional vaccination. In other two projects that I have worked on, you mentioned them, pool testing. So here we try to find a way how to better test patient with using less resources. So and then also production number. In this case, this is a component where we try to look at the incidence data and then try to understand how well we are controlling COVID in our patient compared to general population, and then how we are doing in terms of policies that are embedded in controlling this disease.
Now let me come to pool testing. Actually, I believe some of our members of our audience may want to Google pool testing. And actually, when you do it, I did it a few weeks ago, it's mainly about testing swimming pools, you know. So, and so, what actually is pool testing in the context of COVID-19?
So, what pool testing is a way to actually gather a bunch of samples and test them in a single shot. So imagine that you have 10 patients. Now, one way you can do it traditionally is test every single patient. So in this case, you will do 10 patients. Pool testing is saying basically, depending on some of the characteristics of the disease and the testing device you're using, you can just lump all those 10 patients into one sample and then test that. So in this case, you can see if the patients are negative, you only perform a single test there instead of doing 10 tests in order to find out that all those 10 individuals are negative. But however, if the patient that you lump in those groups, one of them are are positive, right? Then you test every individual one. But if they're negative, you only have performed a single test. So pool testing allows you to kind of reduce the number of tests that you perform, speed up the testing procedure, and then without basically spending too much resources on doing the surveillance.
Yeah, okay, no, this is actually a good explanation, but now let me think this through a bit. So you said, well, you have samples from 10 patients, you put them together and then you test. And if the test for this pool or this group is negative, then you can say, very likely none of those 10 subject has COVID-19. But I mean, say you live under circumstances where a larger number of patients have COVID-19. So what's called prevalence in the term of epidemiology. Say you have a prevalence of 20%, so that 1 in 5 is positive on average. I mean, how would that work then?
So what we did on our paper is to look at what conditions that pool testing is more beneficial. So we showed that if you're above 30%, pool testing is less effective. It's actually useless in that context. But if you're below 30, you can, in some sense, you can actually test it. So, but the higher your prevalence increases, the more likely that you will miss some of those patients. So the recommendation is that it's pool test in general is better when you have a lower prevalence, let's say 1%, 2%, like in a single digit, then it's more efficient to use pool testing. But as you increase the prevalence, then you start, even though it can still be, can provide cost saving to you, but you risk missing some of the patient that you were supposed to detect. So that, which is usually due to dilution effect, and then you increase the negative, like the number of negative, false negative in the parlors of epidemiology.
Okay, so I think this makes a lot of sense. Say if only I'm making up an example, if only 1 in 50 patients has COVID, and you pool and you have 50 patients you want to test and group them in 5 pools with 10 samples each, then there might only be 1 pool positive, and then you just have to test all these 10 specimens in that pool separately. But for the other 40 patients, it's actually 4 tests are enough, right?
Exactly.
Yeah,
exactly.
Okay. No, this is actually a really smart method. Is this method around for a while, or did it just come up with COVID-19?
So actually the method has been around more than 30 years. So they have used them in blood, like a blood bank, where people basically use in order to minimize the sample test, because in this case the prevalence of some infection in your blood in this blood bank is much lower, so it's more efficient there. They have used them in the HIV testing in the early age of HIV, and I've also used them in some other STD, infectious disease. But now it's becoming more discussed during COVID because of, historically we were behind testing availability. So in a sense, pool testing is having a reemergence of cancer, even though a few years ago it was kind of tame.
That's interesting. I mean, is pool testing actually done somewhere? Is it, say, FDA approved? What's the status there?
A few months ago, like around the same time we had a paper, FDA actually had a recommendation on pool testing, and that recommendation was don't do more than 5, right? And that makes sense because the prevalence in some area were much higher than what we recommended that you should do the pool testing under. So it's used in that for COVID. In a sense, there was a recommendation regarding pool testing. But some other, usually it depends on the laboratory that are testing it and how well they are able to handle some of the drawbacks, particularly the dilution effect that will eventually emerge as you pull more people together.
Okay, understood. Now, you also conduct other research in the COVID world. You're dealing with estimating reproduction numbers. Can you just remind me, and maybe also some in the audience, what actually is the reproduction number? What is it used for? What kind of research are you conducting on the reproduction number?
A reproduction number is actually a measure that public health officials use in order to see whether or not the disease is more likely to spread out quickly, or it can be suppressed, right? And generally, when it's above 1, it's considered that the disease has a higher likelihood of spreading through the population and being maintained in the population. When it's less than 1, it means it can be suppressed easily. But what it actually means is that it's the expected number of an individual person, infected person, that can spread the disease to other. So example, you can look at it, let's say if I'm infected, right? And I infect 2 people. So then I will have a production number of 2, because during the course of my disease, I'm able to spread the disease to 2 people. And if each of those individuals also spread the disease to other 2 people, and so on, then eventually what you will see in average will still have a reproduction number of 2. Now, you can easily see that if I infect 1 person and each of those individuals infect 1 person, with some probability that one of those patients will not infect anyone, and then the disease can disappear, right? So it will stop once all the previously infected recovered. So that's what it measures. Yeah.
And say, for example, a reproduction number of 0.5, would it mean actually that 2 people on average would infect only 1. Is this correct?
So no, so what? Because it's a game of average, right? So it means that if you average, yes, those 2 people, so it's possible that one person infected one person, right? But the other person did not infect anyone.
Okay, yeah.
Right.
Yeah.
So therefore, they have an average of 2.5. So yeah.
Okay, no, this is very helpful. What were the results of your research into the reproduction number in dialysis patients?
So what we found is that if you compare the dialysis population with the general population, the reproduction number overall were much lower in dialysis population than general population. Of course, there are some sporadic point in time when this is flipped, but overall, dialysis population were able to control better the reproduction number, even though you also see that the trends, the shape of the reproduction number follows each other. They're similar, almost identical, but the scale, the level of them are actually different.
Why do you think is this the case that the reproduction in dialysis patients would be lower compared to the general population? I mean, I'm thinking like, could it be that dialysis patients are less outdoors because they're physically, the mobility is on average less, or that they wear more masks, or?
So it's combination of factors. One possibility is what you actually put your finger on, is that dialysis patients are less mobile, right? They move around less around the town, so they're less likely to be exposed to the disease. And second, they test frequently, right? If you test frequently, you can put in place certain requirement, right, that actually minimizes the reproduction number. So, for example, dialysis patients in clinics are required to wear a mask all the time. So that itself actually minimizes the spread of the disease within the clinic. But also, if that becomes a habitual practice among them, they also are less exposed to being infected. So those are the one or two factors that can explain why the reproduction number in dialysis population much lower. But I think the culprit is mostly the lifestyle.
Very interesting. Now let's change gears a little bit. I mean, you do, of course, a lot of work in COVID, but there is also other projects where you would use mathematical models. Can you say a few words about those projects?
Yeah. So one of the projects that I'm actually excited about is a vascular classification, and this project is pathophysiologically, it's a really complicated process. So we use mathematical model, mainly data-driven approach to trying to understand the physiology of vascular classification, and at patient level trying to understand the drivers of vascular classification with each patient. So this is, in this case, we use combination of mathematical modeling, understanding of physiology, and also data-driven approaches like a machine learning. The other project that I am working on is detection of intermittency in oximetry data. So like oximetry data measure the level of oxygen in your blood. So we try to look at it and to see what kind of pathologies are associated with intermittency. So it can be intermittent hypoxemia, it can be outside the renal wall, it's fetal hypoxia can generate themselves as intermittency. Basically, this allows us using mathematical model, mainly from a dynamical system, to trying to understand this behavior in that data set.
This is very interesting. You look into oxygen saturation in the arterial blood and in signals, and try to delineate the underlying pathology. That's right?
Yes. So because some patients may have a different pathology, and that can actually be seen in data. In like some patients, let's say if you're they have apaxia, like hypoxemia, then the oxygen saturation can be below 90, but that doesn't necessarily mean they should show those intermittency in their data set. But some patients would show intermittency, or some patients, even though they have oxygen saturation that's above 90, they can still show this intermittency. So this allows us to actually detect those those patterns in the data set in one either retrospectively or in real time by just following some of these signal in the data set.
So you also work on acid-base metabolism and you on bone and I don't know if you have there may not be enough time to go into any details here, but what I find so impressive that you as an applied mathematician, I mean, you work on so different projects from COVID pool testing to acid-base metabolism, to oxygen pattern, to bone metabolism, and so on. So this is quite remarkable. And I think in what I read in your CV in the past, you would work on nanotechnology, on questions that came up in that area, or biochemistry.
I was fortunate enough to be exposed to different fields from undergraduate because my original training was actually in engineering. And I was trained as electrical engineering, but then I worked in nanobiotechnology, particularly mostly developing biosensors to detect environmental pathogens in the environment. And we also developed mathematical method to kind of differentiate which design that you need to incorporate in order to have the most effective configuration in terms of this biosensors device. And I worked in the optofluidics, so this is using light to move fluid around and at the same time doing PCR within the droplet. So I was fortunate to be exposed to these different background.
Do you think that there are actually problems specifically in medicine or biology, that will most likely remain intractable to mathematics? Or do you think that mathematics in principle has all the tools that would help us to understand this? I mean, one of the reasons why I'm asking is, and I'm a total layperson here, I read that certain mathematical developments were absolutely necessary to move forward in quantum physics. And I'm wondering are there certain mathematical new methods possibly even necessary to move into certain areas of this highly complex field of mathematics, of biology? I mean, and so what's your thoughts around that?
I would say definitely. So I'm a believer that actually only imagination can limit the ability to solve certain problem. So I wouldn't say it's necessarily intractable for mathematics. I would just say is like how in the hand of who these tools are in, and the problem they are interested in, either in the availability of data set, right? If we understand some basic understanding of the biology, or if we can measure certain quantity. Once we can measure some quantity, I think that is the limitation. So, for example, if you look at topology, right? Topology was completely, like maybe 2 decades ago.
Do you want to say a few words what topology actually is?
So topology is basically is analysis of sets where they measure shapes and structures, right? So it's in a sense topological structure of properties. 20 years ago, it was purely in the realms of pure mathematics. But now it's actually being used a lot in data science, where you're trying to understand the shape of the data, and it's being used to detect fraud in financial systems. It's used to detect or differentiate different type of diabetes in patients. It's used to treat to basically detect different form of cancers or shape of cancers. So, which initially was previously unknown, but in the hand of the right person with the availability of computational power and some other understanding of the system, mathematics can actually help transition to be used in a field that that was never taught to be used in before. So I think because I'm a little bit optimistic in the use of mathematics, I wouldn't say there aren't any field that will be intractable. It's just a matter of how the type of the data we have and the tools and the skill set of the modeler, how they implement them.
Now, this is actually something really interesting. You said that topology, which was originally just in the realm of pure mathematics, moved over to applied mathematics and is now used in data science. I mean, on the other hand, there is the possibility that certain problems are looked at. Like I think about, when a German mathematician, very famous one, Gauss, was working on measurements, and because there was a problem in the real world, this gave rise to new fields of pure mathematics. Or do you think is this more a two-way street, or do you think that that it's primarily pure mathematics that then inspires applications in the real world, or is it real world questions that are then leads to developments in even with pure mathematics?
So it can go both ways. I'm a little bit biased towards, because my training is applied math, even though I use some like a pure math in my work. So I'm biased in that sense, but generally it goes both ways. Sometimes some problems are actually motivated, like pure mathematical problems are motivated by applied question. And then the pure mathematicians can then go and say, okay, what are the relationship between this structure, right? Now it will go into the realms of purely abstract entity. Now it can also go the other way around, where the problem started purely, the problem started as a pure entity. So you can see this in the contribution of Hardy, right? So this is a renowned German mathematician who believed that it would never see his work applied in real world. So in a sense, he even prided himself of never, his contribution never hurting or helping humanity, right? However, a few years later, his contribution will actually, some of his contributions started to appear in genetics, right? So this, and to the point where they even have a principle named after him, like Hardy-Weinberg principle, where they try to look at the distribution of these the interaction between recessive and dominant trait. So it can go both ways sometimes.
That's interesting. I mean, and I recall, when, whatever, 30 years ago, when suddenly chaos theory was in. Everybody would talk about chaos theory, and it was just it was something that was, or maybe still is, very difficult to understand. Is there actually applications of chaos theory in medicine or biology?
Definitely. But first I will just describe what chaos means, right? Chaos, it's any dynamical system that looks random or irregular, but underneath it has very well behaved deterministic laws that govern this transition, but it's more sensitive to initial condition, right? So if you look at it from that perspective, if let's example, the father of chaos used to say, the future, my current present predict my future, right? But my approximate present does not predict my approximate future, right? So in a sense, what this means is that if I start with two different initial conditions that are very close to each other, then over time, if you run them, they will actually diverge from each other. So they will have completely different features, even though they almost started at the same point. In medicine, so initially in the early years of chaos, what you were talking about, that people started trying to say how to apply the concept, right? But however, I guess there was more bust in the early phase of the application of chaos. But if you can actually look at it without calling it chaos, we do use the technique in our systems. Example, I talk about intermittency, right? How to detect these abnormalities in oximetry data set, right? So where some signals will give you, they'll show you something. It seems that they're random, but they're not in theory random phenomena. They actually have certain properties, certain structure. So in that particular case, we are in fact using concept for chaos theory to actually try to detect them, even though we don't necessarily call them chaotic tools. But there are chaotic tools. Either you use recurrence method, or we use entropy to try to detect them. So there are ways we can use the concept of chaos, but without really using the term chaos.
Could it be that a random event results in this very small difference in the initial condition and then actually gives rise to chaos as you described it?
Yes. So you can. So if a system is a chaotic, right, you can even like measurement error. An example, if you the tools that you use can probably slightly perturb it. The initial condition, even though in terms of precision they're identical, can actually result in trajectory moving apart from each other at certain time and point.
No, really, thank you. I mean, oh my God, I could continue with this conversation for hours. I find it just so fascinating to see what can be done with mathematics. Mathematics is so much more than what I guess many of us have learned in school, and it actually can be applied to the betterment of patients and to people. So I really thank you so much, really, Alhaji Dr. Cherif, for this conversation. It was a tremendous pleasure having you here. And I'm looking forward, of course, to many more conversations outside of this broadcast. Thank you.
Thank you. Thank you for having me, Peter.
Thank you for joining the Renal Research Institute for this episode of Frontiers in Kidney Medicine and Biology. We invite you to engage with us on our social media channels and look forward to seeing you again soon for the next episode of Frontiers in Kidney Medicine and Biology.